بسم الله الرحمن الرحيم والصلاة والسلام على أشرف المرسلين سيدنا محمد وعلى آله وصحبه أجمعين. نقدم لحضراتكم الجزء الثالث من محاضرات العامة لمؤتمر العلوم الصيدلية كلية الصيدلة جامعة أسيوط. لدينا في هذا السيشن أهم محاضرين. المحاضر الأول بروفيسور دكتور محمد عباس إبراهيم، Department of Pharmaceutics, Faculty of Pharmacy, Al-Azhar University, Asut, Egypt. Kayal Chair for Pharmaceutical Industries, Department of Pharmaceutics, Faculty of Pharmacy, King Saud University, رياض، المملكة العربية السعودية. المحاضرة بعنوان Nanoparticle Based Drug Delivery Systems, Different Technologies and Potential Applications.
And the second lecture will be presented by Professor Mahmoud El-Sabahi, Associate Professor, Department of Pharmaceutics, Faculty of Pharmacy, Asut University, Asut, Egypt. And the title of his presentation will be Engineering Advanced Hemostatic Devices by Exploiting Nanocarriers with Tunable Characteristics to Address Global Health Challenges.
The third lecture is by Dr. Muhammad Kamal Hussein, the Whale City for Sciences and Technology Center for Genomics, Egypt, Ecology Department, Faculty of Science, Port Said University, Egypt. The lecture is Role of microRNA 31 in Gynecological Cancer, when microRNA with opposite functions.
And the last presentation will be presented by Professor Hassan Azazi, Professor and Chairman, Department of Chemistry, School of Science and Engineering, the American University in Cairo, Egypt. The title of his presentation will be Top CRISPR Startups.
نتمنى لكم حظا سعيدا في الاستماع للمحاضرات ومناقشتها.
Assalamu alaikum wa rahmatullahi wa barakatuh. First of all, I would like to thank the conference organizing committee to invite me to deliver a lecture at this conference. My wish is that this corona pandemic will end and the world return to normal life, and we will meet at the next conference without social distancing. My lecture today will focus the nanotechnology and the applications of nanotechnology in drug delivery and drug targeting. The title is Nanoparticles-Based Drug Delivery Systems, Different Technologies and Potential Applications. As we know, non-organized living things are abundant in the world. For example, bacteria cell is about 1000 nanometer, virus is about 100 nanometer, and glucose molecule is about 1 nanometer. Nanotechnology is the science that deals with the creation of materials of a scale of 0.1 to 1000 nanometer. The application of nanotechnology in several areas is growing, including agriculture, communication, paints, and of course, medicinal application. The major goals of the nanotechnology in pharmaceutical and medicinal fields could be summarized as enhancing bioavailability by enhancing drug solubility and enhancing its dissolution rate, targeting the drug to a specific area or specific organ, controlling drug release rate, and the use of nanoparticle-based system in tumor imaging and identification of malignant tissues. Of course, wide range of application of nanotechnology in medicine is growing. The pharmaceutical nanoparticles could be categorized as nanonized drug particles or API nanonized particles and polymeric nanoparticles. Polymeric nanoparticles are prepared by dispersing or encapsulating the drug in the matrices of different types of polymers. This category includes hydrophilic or hydrogen nanoparticles, biodegradable nanoparticles, and hydrophobic nanoparticles. While the nanonized drug particles are prepared by reducing the drug particles or initiating the drug molecules to the nano size range so as to enhance the drug bioavailability and dissolution rate. Polymeric nanoparticles, as I said before, they are prepared by dispersing or encapsulating drug into the matrices of different types of polymers. Hydrophilic polymeric nanoparticles are called hydrogel nanoparticles. They are prepared from natural synthetic polymers such as polyvinyl alcohol, polyvinyl pyridone, alginate, and others. These particles combine the properties of hydrophilicity with the properties of nanoparticle attribute as the very small particle size. There are several potential applications of the hydrophilic or hydrogel polymeric nanoparticles in biological and medicinal fields. These nanoparticles have high affinity to absorb water due to the presence of hydrophilic groups on the surfaces of their molecules. For example, abraxane is an example of the marketed hydrophilic nanoparticle containing the anti-cancer drug Docetaxel. These nanoparticles, or this nanoparticle-based system, is about 130 nanometer in size, and it has several advantages over the conventional marketed paclitaxel product in terms of reducing toxicity and dosing flexibility. Biodegradable nanoparticles are nanoparticles prepared by dispersing the drug molecules or encapsulating drug molecules into the matrices of biodegradable polymers. These polymers, upon degradation, they degrade into biocompatible and biodegradable degradation products. These biodegradable nanoparticles have long half-life, high entrapment efficiency. They are used for site-specific targeting, and they have the ability to permeate through skin, and therefore they are widely used for transdermal application. Biodegradable polymeric nanoparticles include, for example, starch nanoparticles, calcium nanoparticles, polylactic acid and polylactic glycolic acid nanoparticles, polycaprolactone nanoparticles, for example. The third category of polymeric nanoparticles is hydrophobic nanoparticles. These nanoparticles are prepared using hydrophobic or fatty nature polymeric materials. This category includes solid lipid nanoparticles, self-nanoemulsifying drug delivery systems, liposomal drug delivery systems, silicon nanoparticles, and gold nanoparticles. These nanoparticles, the hydrophobic nanoparticles, are used routinely in water purification and detoxification, in the protection of incorporated labile drugs, in controlling the drug release rate, and in targeted drug delivery systems. There is an example of hydrophobic nanoparticle marketed product, which is liposomal doxorubicin formulation available in the marketed in several markets. This product have several advantages over the conventional drug in terms of reducing cardiac toxicity, especially in elderly patients. The second category of nanoparticulate drug delivery systems is the nanonized active pharmaceutical ingredient particles. These systems are prepared by reducing the coarse particles into nano-size range or initiating the aggregation of molecules to aggregate to form nanoparticles. Nanonization of API could be carried out via two different techniques, including top-down technique and bottom-up technique. Top-down technique is based on the reduction of particle size of the micro size or coarse size particles to the nano size range, while the bottom-up technique is based on the initiation of aggregation of drug molecules to form nano-sized particles. This could be carried out via precipitation or spray drying or several methods. As I said before, top-down technique is based on the reduction of particles to the nano-size range, which could be carried out via milling or grinding and high-pressure homogenization, while bottom-up technique is based on the initiation of drug molecules aggregation to form nanoparticles by supercritical fluid, spray drying into liquids, rapid expansion of supercritical solution, and precipitation with compressed fluid and solvent. Particle size reduction could enhance the solubility of API through different mechanisms, including surface area increasing the number of high energy sites, and increasing crystal defect with the changing the crystalline nature from crystalline to amorphous state, and by reducing the drug melting point. These mechanisms, one or two mechanisms or more, can result in enhancing drug dissolution rate and therefore enhancing drug bioavailability. However, the nanoparticles are highly energetic particles due to the high surface area of these particles, and therefore the drug or the nanoparticles tend to minimize their surface free energy by being close to each other, and therefore several stability issues occur in nanoparticulate-based drug delivery systems. These issues are concerned with the aggregation of particles, including sedimentation, aggregation, and crystalline transformation from amorphous to the crystalline state. Therefore, the addition of stabilizers to the nanoparticulate drug delivery systems is a crucial point. Stabilizers are added to prevent the aggregation of the nanonized particles and to enhance the stability of nanoparticulate system. These stabilizers may include polymers such as polyvinyl pyrrolidone, hydroxypropylmethylcellulose, hydroxypropylcellulose, and may include non-ionic surfactants such as twins and polyuronics. Also, ionic surfactants could be used as electrostatic stabilizing agents, including sodium lauryl sulfate and sodium deoxycholate. Both stabilizers are acting via different mechanisms to stabilize the nanonized drug particles. Polymers, for example, act by steric stabilization, in which the chain of the polymer attached to the surface of the nanoparticles and make bridging between the two adjacent nanoparticles. This will result in reducing the aggregation and preventing particles from being to each other. On the other hand, electrostatic stabilizers are acting by covering the surface of nanoparticles with same charge, and therefore repulsive force will increase, and therefore the drug nanoparticle stability will be enhanced. In the coming slides, the next slides, I will show some examples of the nanotechnology research in our department, of Pharmaceutics, King Saud University, in Kayal Chair for Pharmaceutical Industries. For example, I will show an example of enhanced drug dissolution rate of some statins, including Rosuvastatin, calcium, and simvastatin, enhanced corneal permeation and residence time of the antimycotic drug Fluconazole, improved dissolution and cytoxic activity of Docetaxel, anti-cancer drug, minimization of pioglitazone hydrochloride fluctuations in plasma concentration by minimizing the precipitation that occurs upon changing the pH from stomach to intestinal pH, which results in precipitation of the drug and therefore will be accompanied by erratic absorption and reduced drug bioavailability. First example of enhanced dissolution rate of statins, we choose Rosuvastatin calcium, which is big, high molecular weight statin. We prepared Rosuvastatin calcium nanoparticles by using planetary ball milling or wetting milling technology. We optimized the formulation and process parameters to obtain the optimum conditions to get the optimum formula of drug nanoparticles. The optimum conditions for nanonization was using milling ball size of 0.1 millimeter, milling speed of about 450 RPM, and drug to solvent or milling to solvent ratio is 0.25 to 1. We used here 10% polyvinyl chloride as a nanoparticle stabilizer. The obtained results of the optimized drug formula showed enhanced initial dissolution rate of the drug and enhanced dissolution efficiency after 1 hour in comparison with the untreated Rosuvastatin calcium. In addition, the prepared nanoparticles showed nano size range of about 160 nanometers. After preparing the nanoparticles and freeze drying, we try to compress the prepared drug nanoparticles of Rosuvastatin into directly compressed tablets. And we found that the drug exhibited a slow dissolution rate from the tablets containing drug nanoparticles in comparison with the tablets containing untreated drugs and the tablet containing the commercial product crystal. In addition, we found that the disintegration of the tablet containing nanoparticles was very slow in comparison with the tablet containing the commercial tablet and the tablet containing the untreated drug. When we analyzed the scanning electron microscopy, analyzed the crushed tablets with the scanning electron microscopy, we found that the prepared nanoparticles are adhered or were adhered to the surface of the tablet filler abyssal, and here is the nanoparticles. This might be due to the binding effect of the nanoparticle stabilizer polyvinyl pyrrolidone. Therefore, the incorporation of rosuvastatin calcium nanoparticles into directly compressed tablets was difficult due to the binding effect of nanoparticle stabilizers that resulted in slow disintegration and slow drug dissolution rate. Therefore, we changed the incorporation of the drug nanoparticles from tablets into capsules, and we compared the dissolution of drug nanoparticles from capsules in comparison with the dissolution of untreated drug with or physically mixed with polyvinyl pyrrolidone. There was significant enhancement of the drug dissolution rate from hard gelatin capsules in case of drug nanoized particles. In addition, the pharmacokinetic studies from capsules containing rosuvastatin calcium nanoparticles showed improved pharmacokinetic parameters in terms of short Cmax and high value of area under the curve in comparison with the capsules containing untreated drugs or the capsules containing untreated drug physically mixed with polyvinyl pyrrolidone, which is nanoparticle stabilizer. We published 2 papers concerning the formulation of rosuvastatin calcium nanoparticles, and in these papers we discussed in details the formulation and process parameters that might impact the behavior of rosuvastatin from these nanoparticles, and we try to optimize the planetary ball milling conditions for good or for small particle size drug particles, in addition to enhanced dissolution rate. The second example in our lab was the enhancement of corneal permeation and corneal residence time of the antimycotic drug Fluconazole. The ocular delivery of Fluconazole showed low bioavailability, less than 10%. This might be due to the impermeable corneal epithelium membrane and rapid corneal elimination as well. Therefore, we formulated fluconazole nanoparticles and incorporated these nanoparticles into ocular gel. The drug nanoparticles, fluconazole nanoparticles, were prepared by the anti-solvent precipitation method, which is one of the bottom-up method, and we used several stabilizers at different concentrations to screen the effect of these stabilizers on the drug nanoparticles. These stabilizers include hydroxypropyl thiacellulose, polymeric F127, Colicot, sodium lauryl sulfate, and others. And the results showed that the nanoparticle formula F3 that stabilized by 5% polymeric F127 exhibited the best attributes in terms of small particle size, about 352 nanometer, small PDI, and reasonable or acceptable zeta potential -18. This nanoparticle formula was incorporated into the ocular gel prepared from carbopol 934, and the results showed that enhanced drug release from carbopol gel in case of using drug nanoparticles in comparison with the untreated drug. The drug release was performed using ex vivo study by using goat cornea. Also, the pharmacokinetic studies of the nanoparticle formula prepared by Pluronic F127 was studied for ocular pharmacokinetics in rabbits, and the results revealed that the drug exhibited high or improved dissolution rate. Improved pharmacokinetic parameters in terms of reduced elimination rate constant and increased area under the curve. This work was part of a master degree presented in 2018. The second example is the enhanced dissolution rate of cytotoxic drug. We tried to enhance the cytotoxic activity of the anti-cancer drug Docetaxel by formulating drug nanoized particles using anti-solvent precipitation method. We used two stabilizers here, hydroxypyramidicellulose, low viscosity grade, and the sodium deoxycholate. The results showed that the nanoparticle formula prepared by using 5% sodium deoxycholate showed or exhibited very small particle size, about 80 nanometer, with acceptable zeta potential value. In addition, the drug exhibited high dissolution rate from nanoparticles stabilized by 5% sodium deoxycholate in comparison with other sodium deoxycholate concentrations and with the untreated drug. In addition, the drug exhibited high cytotoxic activity in case of the nanoparticles stabilized by 5% sodium deoxycholate with the use of low drug concentration. From the untreated formula. In addition, there was a precipitation after specific time due to the low solubility of the drug in the pH, in the dissolution pH. The high standard deviation values indicate the very difference in the dissolution points. However, the dissolution rate of the drug from the freshly prepared nanoized particles was very high, significantly, and the dissolution after 3 months storage was also higher in comparison with the untreated drug, but with little reduction in its dissolution rate due to the storage. This might be due to increase the size of the nanoparticles, which resulted in slowing of the drug dissolution rate, but still higher than the dissolution of the untreated drug. The bioavailability studies for the optimized pioglitazone nanoparticles was significantly showed that there is a significant difference between the pharmacokinetic parameters in case of optimized nanoparticles in terms of high Cmax and increased area under the curve in comparison to the pharmacokinetic parameters of untreated drug. This might be due to reducing the fluctuation of the drug absorption and by minimizing the precipitation of the drug from or upon pH change from stomach to intestine, and this was achieved by the nanoization of drug particles. We published two chapters concerning the application of nanotechnology in drug delivery. The first chapter was published in the book published by Elsevier. The book title was "The Surface Chemistry of Nano-Biomaterials, Applications of Nano-Biomaterials." The drug chapter titled Nanotechnology from Particle Size Reduction to Enhancing Aqueous Solubility. This was published in 2016. Recently, in 2020, we published another chapter in the book published by Springer. The book title was Nanopharmaceuticals, Principles and Applications, Volume 1. Our book chapter carried the title of Applications of Nanopharmaceuticals in Drug Delivery and Targeting. There are several marketed products that are based on the nanoparticulate drug delivery systems. These products are mostly prepared by top-down media by size reduction, including Ripamune, Ement, Tricor, Fenofibrate, Griseofulvine. Some of these products are prepared by spray drying or bottom-up technology. However, the applications of nanotechnology in drug delivery are still growing. I would like to thank my colleagues, Professor Faris Al-Anazi, supervisor of Kayari Chair for Pharmaceutical Industries, Dr. Ahmad Abdul Fattah Abdul Latif, the Professor of Pharmaceutics in Al-Qazim University and Al-Azhar University, our professors from the female section in King Saud University, Dr. Dua Al-Shura and Dr. Amal Al-Saif, and the master students, Bandar Al-Maliki and Yara Al-Qahtani, and would like to thank you for your attendance. شكرا جزيلا.
السلام عليكم. طبعا انا يسعدني يعني ويشرفني ان انا يعني يبقى ليا كلمه في المؤتمر بتاع جامعه اسيوط، وبشكر اللجنه القائمه على المؤتمر والمجهود الكبير جدا اللي عارف ان هم بذلوه علشان ينظموه قبل كده، ونتيجه لظروف كورونا ان هو ياجلوه ويعيدوا تنظيمه تاني. فطبعا الشكر لجميع اعضاء اللجنه وعلى الاخص يعني عميد الكليه الدكتور محمود شيحه ووكيل كليه الدراسات العليا الدكتوره جيهان نبيل. شكرا جدا على مجهودهم. My talk today is on engineering advanced hemostatic devices by exploiting nanocarriers with tunable characteristics to address global health challenges. Bleeding is, as we all know, is responsible for about 35% of pre-hospital death and over 40% of death within the first 24 hours. Hemorrhage is considered as the leading cause of death in traumatic injuries and the fourth leading cause of death in the United States, with a total cost of $671 billion in 2013. Usually, there are several methods for mitigating severe bleeding, either in accidents or in battlefields or others. Most of them depend either on surgical suturing or the use of electrocauterization or the use of hemostatic dressing. Hemostatic dressings have the ability to reduce the morbidity and mortality associated with bleeding via the early control of hemorrhage. Usually, there are different shapes that can be designed depending on the medical application. So, for example, they can be very small pieces to control dental or nasal bleeding. They could be larger pieces to control acute hemorrhage after traumatic injuries in battlefield or in accidents, or it could be of moderate size as in the first aid kits. And these are some examples for different hemostatic dressings of different composition and different natures, either biodegradable or non-biodegradable, as we will discuss later. And all of those hemostatic dressings are available in the market. Kaolin is one of the hemostatic agents that has been used since very long time, and it is actually hydrated aluminum silicates. It acts by absorption of water and rapid concentration of platelets and clotting factors to catalyze the rapid clot formation. Although the efficiency of kaolin in controlling the acute hemorrhage, some of the main drawbacks associated with kaolin is the exothermic effect after application to the wound area, and also flooding at the bleeding site. So these are the main two limitations for the use of kaolin as a hemostatic dressing. One of the most commonly used hemostatic dressing is quick clot combat gauze. It is a rolled gauze impregnated with kaolin. It is almost carried by all branches of the US military to control acute hemorrhage after injuries. It is licensed for the temporary external control of traumatic bleeding, although there are several literatures are using it for other purposes to control hemorrhage from other sites. The advantage of the use of gauze that is impregnated with kaolin as compared to the free powder is that they can reduce the exothermic effect. However, the main limitations of quick clot combat gauze is the moderate efficacy. It's not very efficient in controlling the bleeding, and it is non-absorbable. We will discuss this later, but just to explain to you the meaning of non-absorbable, it means that after stopping or controlling the bleeding, it has to be removed from the injury site. Chitosan is derived from chitin, which is natural polysaccharide, occurs in several crustaceans. It is obtained from chitin via deacetylation, and in vivo it should undergo biodegradation into N-acetylglucosamine and glucosamine. The mechanism of action of chitosan that acts by accelerating blood clots by interacting with platelets and coagulation factors. It has intrinsic bioadhesive and antimicrobial properties, and it is the active ingredient in several commercially available hemostatic dressings such as Hemcon and Chitodine and Celox. The chitosan powder itself is non-bioabsorbable. It must be removed from the wound site, and it forms aggregates in aqueous solution. The idea of the projects that we are working on is that if we could increase the surface area of chitosan, for example, via nanoscale assembly, this can increase the hemostatic efficiency and prevent the formation of aggregates in vivo. And so if we prevent the formation of aggregates in vivo, so they can biodegrade over time, so it becomes absorbable dressing. However, the main challenge is that after formation of, for example, of chitosan nanofibers, once they are dispersed or dissolved in solution, they lose the nanoscale features and become just dissolved as chitosan powder. We have used chitosan nanofibers previously coated on gauze, and they have shown exceptional hemostatic efficiency via reducing the blood loss and reducing the time to hemostasis and increasing the survival rate and maintaining normal mean arterial blood pressure when they were tested in lethal injury rabbit models. However, the main problem with the previous system, it was very efficient and it can have application for certain purposes, like for example, to control bleeding in battlefield or in severe injury in hospitals. However, they are still non-absorbable. So if they are used in a small surgical operation or to control bleeding in a small area, they must be removed from the surgical site. So the idea of this project was to convert the non-absorbable dressing into absorbable one. Why? First of all, as we mentioned, the use of absorbable hemostatic dressing can eliminate the necessity for carrier removal and avoid the risk of rebleeding and reduce the operation time needed, which is very useful for several surgical and medical applications. The powdered hemostat, so if we use the powder, even absorbable like dressing, the problem is that they usually float on the bleeding side, which compromise the hemostatic efficiency. So in this project, we could design hemostatic dressings that are absorbable, so they degrade over time, as like second version or an updated version of the non-absorbable hemostatic gauze that we have used previously. So in the table, there are several examples of absorbable hemostatic dressings that are already in the market. Most of it, like such Surgicel or Surgifoam or Edicel or others, and most of them are either comprised of gelatin or collagen or oxidized regenerated cellulose. So again, our project, the idea was to design sacrificial carrier that can allow the assembly of nanostructured chitosan and eliminate the need for further interventional procedures by generating bioabsorbable composite wound dressings. So first of all, we started with cyclodextrin polyester as a sacrificial template. With this one, we add the chitosan, and as we can see here, we observe increased opacity after addition of chitosan, which indicates the chitosan loading, to form a malleable and flexible hydrogel that can be applied to the injury site. Then we removed the template, the sacrificial template, just to understand the assembly of chitosan. And as we can see here, they form, and we will see it in more detail, honeycomb-like nanofibrous mat of chitosan with a nanofibrous diameter of around 9 nanometer. But when we used it to test the in vivo efficiency, we used the whole hydrogel because this layer was only used to study the assembly of chitosan. And as we can see here, they were like tough, non-flexible, so it's difficult to apply to the injury site. When we tested the efficiency of the hemostatic hydrogel, we could observe superior and exceptional hemostatic efficiency, as we will show later in the data that we obtained. So here you can see the structure of the cyclodextrin polyester, and it was very carefully designed to have hydrolytically degradable or cleavable ester linkage to allow for physiological degradability. And the soft linkages also allow the material flexibility because the injury site, I mean, it could be deep, it could be of irregular shapes, so you need flexible materials so they can fit easily into the injury site. Also, the carboxylate groups allow the interaction with the cationic chitosan chains. So here we can see that the macroporous structure of the sacrificial template. When we tested the in vitro degradation, we can see here the cyclodextrin polyester loaded with chitosan could degrade into small molecular weight components. And when they are stored in 1% acetic acid, the degradation is minimal over 7 days. While in phosphate buffered saline, you can see the percent of degradation increase significantly until they are almost 100% degraded after 5 to 7 days, which is very important because we want to make sure that like this degradable template can degrade, leaving the chitosan in the injury site, which also biodegrade over time. So in this way, we leave it in the injury site and we close the surgical site or the surgical operation, which can significantly reduce the duration required for a particular surgery. Then we tested the hemostatic efficiency in acute liver punch biopsy model. We evaluated the time to hemostasis, the amount of blood loss, and the ability to maintain normal mean arterial blood pressure. So the animals were alive throughout the whole experiment, and all values are presented as the mean plus or minus standard deviation of 6 independent experiments, and significant differences between the groups were evaluated by one-way ANOVA. Here, as you can see, this is our samples. So this is the blank hydrogel, and this one is the chitosan-loaded hydrogel, and they were compared with Surgicel and Curaspan. And here is the acute liver punch model. So I'm avoiding the details of methodology, but you make a punch of 1 centimeter in diameter and 0.3 centimeter in depth, and here you apply your formulations. So as we can see here, with the use of conventional dressing, there was drop in the mean arterial blood pressure, while for most other formulations, the mean arterial blood pressure was maintained. And here also you can see that the active bleeding after, without any dressing, and also with the conventional dressing, you can see the active bleeding, while with the use of the commercially available and the chitosan-loaded hydrogel, the bleeding was controlled. We then measured the time to hemostasis and amount of blood loss in rats, rabbits, and pigs. And here, as we can observe, all the tested formulations significantly reduced the time to hemostasis and the amount of blood loss as compared to the conventional dressing, and also as compared to the hydrogel itself. The chitosan-loaded hydrogel was significantly more efficient in reducing the amount of blood loss and time to hemostasis as compared to the commercially available absorbable hemostatic dressing, Curaspone and Surgicel. So these newly developed absorbable hemostatic dressing with nanoscale features proved to be more efficient than commercially available hemostatic dressing. Then the biocompatibility and degradability of the dressing were also evaluated after implantation in the same acute liver punch model after 7 days. So in the control where there is no any treatment, you can see here the histological examination. With the conventional gauze, you can see, of course, so this is the control without any injury, it's normal. Here is with the conventional gauze, you can see there is like significant inflammation and clear damage and necrosis after removal of the dressing. In the hydrogel, there was like incomplete healing, still you can see it. While with the Surgicel and Curaspone and the developed hemostatic hydrogel, especially in the hemostatic hydrogel and the Curaspone, you can see complete healing with no residues, and it looks like normal liver with a small line of cut. So in conclusion, we could load chitosan into sacrificial template that was carefully designed to be flexible and to allow the interaction of chitosan and to permit the assembly of chitosan into honeycomb-like nanofibrous mats of chitosan. And this significant increase in surface area allowed the improved efficiency of the hemostasis. And when they were tested into acute liver punch model, they were significantly of higher efficiency as assessed by either the ability to maintain normal mean arterial blood pressure and reducing the time to hemostasis and reducing the amount of blood loss. We could also demonstrate their biocompatibility and biodegradability. So after implantation into the liver of the rats for 7 days, we couldn't see any residues and we could see complete healing of the injury site. So the future work is to develop strategies that can allow easier method of manufacturing and handling and storage of the hemostatic hydrogels, and to extensively study the preclinical and the clinical safety and efficiency of the hemostatic hydrogels. So at the end, I'd like to thank Professor Dr. Mustafa Hamad. He is a professor of surgery, and he was responsible for the design perspectives and preclinical and clinical evaluation of the developed hemostatic dressings. I'd like also to thank my group at Texas A&M University who participated in the design of the sacrificial template and in the overall design and study. So here, Eric, who has developed the hydrogel sacrificial template. It was for another application, and we could collaborate together to use it for evaluation of the hemostatic efficiency of chitosan. Nari, who studied the scanning electron microscopy to show the honeycomb nanofibrous mat of chitosan. And Professor Karen Woolley, of course, she's a chemist, and she's a pioneer in her area of supramolecular assembly and polymer synthesis. I'd like to thank different like Welch Foundation and the Associated International Center of Nanomedicine and NASA Space Technology and the LSPI at Texas A&M University for support through the infrastructure or through funding. And thank you for everyone, and thanks for your attendance.
السلام عليكم ورحمة الله وبركاته. أهلا بحضراتكم. اسمحوا لي في البداية إن أنا يعني أشكر القائمين على إدارة المؤتمر وتنسيق المؤتمر وإدارة جامعة أسيوط، وبقيادة طبعا مع الأستاذ الدكتور طارق جمال رئيس الجامعة، ومع الأستاذ الدكتور أحمد المنشاوي نائب رئيس الجامعة. Also allow me please to extend my thanks to Professor Sheha, the Dean of Faculty of Pharmaceutical Science, and Professor Jihan, the Vice Dean, the Faculty of Pharmaceutical Science at Statich University, for the kind invitation. And it's really my great pleasure today to present my data under the title of Role of microRNA 31 in gynecological cancer, and we will see how does one single microRNA show different or opposite function depending on the tissue type. Okay, so first of all, please allow me to go through the content of today's talk. Today's talk will include brief introduction about microRNA and the rationale from which came with our data. I mean, why specifically microRNA and why gynecological tumors, and also we will show some results from 3 different parts. One shows endometrial cancer in MIR31, and other one ovarian cancer, chemo-resistant, and the microRNA 31. And finally, some brief data regarding this application. Then we will sum up the conclusion and show what's going at the moment in our labs. As an introduction, general microRNAs are non-coding genes. It's a newly discovered generation, like 1 or little bit more decade ago. These non-coding genes have been discovered for their function as a regulator for other coding genes. So their origin is the nucleus from eukaryotic cells, and they are transcribed usually as little bit small sequence, but pre-microRNA, which face some preparation and processing as folding like hairpin, and by the help of different enzymes inside the nucleus, this microRNA face some processing and exported by exportin to the nucleus, to the cytoplasm, and in the cytoplasm it will be diced by the dicer and finally cleaved to single strand, which incorporated into the ROSCa complex to inhibit the function of or regulate the expression of other specific messenger RNA. Therefore, we had a conclusion, general impressions that these microRNAs usually regulate or target specifically some genes. We had much interest to see this one in gynecological cancer because we had worked for the almost 15 years ago with the gynecological oncology group, and this group had much interest with uterine endometrial cancer, and ovarian cancer. Of course, gynecological tumors include a lot, like vaginal one, cervical one, of course, germline subtypes, but we had much interest with endometrial and ovarian first. General talking, these endometrial and ovarian cancer, actually, majority of them had been derived from epithelial origin, which make them very highly responsive to the chemotherapeutic regime. Therefore, the best or standard treatment is depulking surgery followed by chemotherapy. This chemotherapy will include cisplatin, carboplatin, and paclitaxel. Cisplatin is a DNA alkylating agent, and paclitaxel is one of the member, very famous one of the member of microtubule stabilizing agent. Okay, so if these tumors usually are highly responsive, what is the problem? The problem is the almost 30 or 40%, which may relapse, actually represent a very big obstacle for gynecological oncologists. Therefore, long time ago, people generally had used to discover the involved gene in the carcinogenesis process by comparing the cDNA microarray between non-cancer and cancer tissues. However, we had interest to compare the microRNA profile between these tissues, and of course, we could also include the advanced cases or relapsed cases. And unfortunately, we find MIR31 is one of the overdoses between these microRNA profile. This microRNA had been increased during the carcinogenesis. Therefore, we had much interest to investigate further what's its role, and we just cloned this microRNA into CMV promoter expression vector. Then we had been overexpressed this microRNA in endometrial cancer cell line, very well established one named HEC50B, and in relative stress like serum deprivation, we could observe that MIR31 had been increased the clonogenic capacity of the cells, and actually that made our attraction. Why don't we move to the in vivo model? In the in vivo model, we found that very similar data, which means MIR31 actually promoted the growth of these tumors in the nude mice. And of course, the greater the tumor, the higher the expression of MIR31 in these tumors. Therefore, we tried to find out what is the target of this microRNA, which had been upregulated seriously with the progression of the tumors. And we found from the prediction database, like target scan, we found that LATC2 is one of the targets of this microRNA 31. And the complementary sequence had been validated. Then we confirmed again that the overexpression of MIR31 had been associated with lower LATC2, and the opposite is true, means anti-MIR31 had been associated with higher LATC2, and also the clonogenic ability of the cells in which we had been knocked down the LATC2, which shown some enhanced clonogenic ability. So what's LATC2? As a reminder, LATC2 is one of the HIPPO pathway, and one of the majority of the major function of this LATC2 is just controlling the phosphorylation of YAP1. YAP1 is in fact transcription factor. If not phosphorylated, it's translocated into the nucleus. If phosphorylated, it remains outside in the cytosol. Okay, if it's translocated into the nucleus, it drives this expression of cyclin D1 and of course promotes cell cycle. Therefore, we validated this phenomenon in our cellular model, and GFP here shows the successful transfection even with a MOC or MIR31 expression vector, and of course only cells with higher expression of MIR31 show nuclear gap. And this nuclear gap actually associated with higher luciferase activity in the cyclin D1 or CCN D1 promoter. Again, this validates that, okay, miR31 is targeting LATS2, so overexpression of miRs might inhibit the LATS2 and, of course, keep YAP nuclear, and this nuclear YAP promote or drives the expression of cyclin D1 and subsequently cell cycle. Then we move to the endometrial cancer tissues. So we screened for miR31 and we could categorize the tissues we have into high expression of miR31 or low expression of miR31. And fantastically, we found that higher expression of miR31 is associated with lower LATS2 and, of course, higher expression of cyclin-dependent cyclin D1. And the opposite was true. And here in panel B, you can see two representative cases, case A and B, very low expression of miR31 and very high expression of miR31. Of course, low expression associated with higher LATs and lower cDNA, CDN1. And the opposite was true, which means high expression of miR31, lower LATs, and very high expression of ND1. Again, we could categorize it. Of course, we linked the LATs expression with miR31 in these tumors, and again, we could categorize the patient to high risk and low risk, and fortunately, high risk population showed higher expression of miR31, and low risk one showed lower expression. As a conclusion, we could summarize that miR31 can function as an oncogene in endometrial cancer by targeting one of the member of Hippo pathway, and of course we can, diagnostically, we can use this miR31 as a marker for prediction of recurrence or bad prognosis. As a part of this data, today's data is focusing on the other organ, which is ovarian. And working on ovarian cancer, again, the standard regime for treatment is surgery followed by chemotherapy. The majority of these tumors, or ovarian tumors, usually highly responsive, except a limited percentage. This percentage may show either natural non-response or relapse. Therefore, we had much interest to compare the microRNA profile between different cancer cell lines, and also we thought, why don't we develop chemo-resistant cell line against paclitaxel as a standard? Of course, we have other story talking about cisplatin to compare the different mechanisms. Today's data regarding paclitaxel, so we developed two cell lines. These cell lines became truly resistant against the paclitaxel, and we compared the array, the microRNA profile between the two cell lines. Of course, first step, we confirm the resistant mechanism and then profile the microRNA from each cells. And fortunately, we found that microRNA 31 is one of the, between the array data, which highly, the most downregulated one. And we have much interest to see, okay, why this microRNA is doing like this. First of all, we went through the panel of ovarian cancer cell lines in our hands, and we checked the expression of miR31. And also we tried to link this one with the response for IC50 to paclitaxel. As you can see, lower IC50 had been associated with high expression of miR31, and opposite is true here. So these cell lines, which we have in our hands, we just developed from these cell lines that chemo-resistant. Here we can see, okay, after chemo-resistant development, we can see the reduction, significant reduction of miR31 expression. This guides us to why don't, again, we overexpress these miR31 in the resistant cell and see what happens. After cloning of miR31 and establishing 3 different clones with differential expression pattern of miR31, and we could use them, first of all, before application of the drug, in the culture, we observed very fantastic observation or phenomenon. As you can see in these videos, this line is the parental one, so after resistant acquisition to paclitaxel, and this line after the overexpression of mirtazapine. So I will show you here in these videos, as you can see, the motility, and I would say the high motility of and less attachment in the parental cells. This high motility had been reduced. The cells became more aggressive, and of course still dividing, but not highly moving in the patient. However, after overexpression, the cells again restore a lot of morphological features as well as parental. This inspired us. Okay, there is very specific mechanism regarding either cell cycle rate or maybe the aggressiveness of tumor tissues in case of chemo-resistant ovarian cancer cell line. 1274, which is actually one specific inhibitor, pharmaceutical inhibitor, now in the clinical world testing or phase 2 trial in case of some MET overexpressing tumor. And fortunately, mix of paclitaxel and such MET inhibitor have been significantly enhanced the shrinking or the reduction of the weight of the tumor in case of compared with control or even paclitaxel alone. And of course, in such cases, the survival of such animals had been completely changed, which means lower survival had been associated, of course, with paclitaxel treatment and MET31 overexpression. Here we move to clinical samples. So the ovarian cancer tissues had been tested. Actually, this small panel of ovarian cancer tissues, we had just checked the expression of MET31, and in fact, these precious samples are driven from patient before and after recurrence. So this we can see these are sensitive tumors, initial sensitive tumor or primary tumors. This one is resistant, and we could summarize that resistant in general to paclitaxel had been associated with reduction of MET31, and importantly, MET31 high expression had been also associated with longer survival rate. Therefore, we could conclude that MET31, in case of ovarian cancer, it had some role to target MET, and it targets, if it's not targeting MET, it allows or increase the chance for paclitaxel resistance, and thus either MET31 itself or inhibitor to MET may enhance or block or inhibit the chance for a relapse or chemo-resistant or paclitaxel specific resistance. Up to here, we could publish some of our data with our collaborators, and now I'm going to show what's possible application of such finding. We thought, why don't we develop some aptamer, especially in case of ovarian cancer or chemo-resistant ovarian cancer tissues or cells. We thought, why don't we find a way to deliver such MER as an inhibitor of growth or enhancer for chemo response. So we thought about the aptamer. We found that, of course, for those, just a reminder, aptamer are very specific oligonucleotide, either RNA aptamer or DNA aptamer, which can specifically target some molecule like proteins or peptides. So we found that people already developed the MUC1, very specific aptamer, and this one was applicable in case of ovarian cell lines. Therefore, we checked the expression as a first step of MUC1 in our panel of ovarian cancer cell line. and we found it's clearly expressed, and especially in our cellular model, KF and Docetaxel cells. Therefore, we thought, why don't we use this mucin-1 aptamer, and of course label with dye like Cy5, and the other side with biotin just to keep stability, and we just merge the sequence with mucin-1 sequence, and of course one specific one as a control. The first step was to see if this aptamer can be delivered into the cell by the help of mucin part or not. As you can see here, the red line indicates the aptamer molecule, Cy5, and we can see the internalization is clear here in the merged photo. And also you can see in such a video that time-lapse fluorescent video shows internalization of the aptamer, which is clearly internalized into the cytosol compared with this. Okay, again, further mechanistic study or molecular study, we confirmed that higher concentration of aptamer had been associated with lower MET expression, which indicate possible response, and of course such kind of work is right now running in our laboratory to validate this finding. However, the current or even future thought outside is, why don't we think about the epigenomic regulation of this microRNA? For that purpose, we again compare the parental and resistant cell in terms of near profile, but we just added also a panel of chemoresistant cell, but treated with epigenomic modulator like JAK inhibitor or DNA-methyltransferase inhibitors, and to see if mainly the role of or the possible downregulation and again upregulation of MIR or whatever molecule. Okay, up to here, I'd like to thank my team, STDF and City for the Fund, British Council, and also ASRT. And thank you very much for your attention, and I'll be glad to receive any question from your side. Thank you so much.
Assalamu alaikum wa rahmatullah. It is my great pleasure to participate in the Asyut University 12th International Pharmaceutical Science Conference. First, I would like to thank Professor Dr. Tariq Abul Fadl for his kind invitation to me to participate in this conference. The title of my presentation is Biosensors for Detection of Biomarkers. My name is Hassan Azazi. I'm a professor and the chairman of the chemistry department at the American University in Cairo. My lecture outcomes include five outcomes, which are describing the significance of using biosensor for real-time monitoring of disease biomarkers, understand the basic structure of a biosensor, listing the criteria of a successful biosensor, become familiar with the selected state-of-the-art biosensors, and drafting a roadmap for designing a biosensor. يعني الحاضرين النهارده ان شاء الله في نهايه المحاضره اللي حوالي 20 minutes هيقدروا ان شاء الله ان هم يجاوبوا على الخمس اهداف ديت او يقدروا يقولوا معلومات عن الخمس اهداف ديت المحاضره. Biosensors are very important for monitoring patients, especially for remote health monitoring. يعني the current system or the current healthcare system aims to help people to live as long as possible a high quality of life. This will be enabled by the use of biosensors, which are small devices, portable devices that patient carry with them in their household and in their environment without having to be hospitalized. The biosensors will detect and monitor different disease biomarkers in real time and in a continuous manner, and send this data to the health facilities. And in case that the patient, will display a danger level of a particular disease biomarker, this information will be sent to the health facilities for an emergency vehicle to be dispatched to the patient, and also alerting the family members and loved ones so they can start moving toward this patient, and also the clinician who is following the patient will be also alerted to the situation. So by the use of different biosensor, information will be sent from the patient to the health facilities, and then the different health facilities will intervene with the patient. What is a biosensor? It is a device that consists of two main components: a biological sensing element and a transducer, which converts the data into electrical signal. طبعا, the most important and the most famous biosensor is the glucose biosensor. So this is the two components of the biosensor. The most important one is the biological sensing layer. So this is a biological entity which can recognize the analyte or disease biomarker. طبعا, it can specifically detect the disease biomarker, but not other molecules in the biological specimen. Then binding of the analyte to the biological sensing layer will result in a signal that will be sensed by the transducer and converted into a signal that is displayed on the screen of the biosensor. The biosensing element could be an RNA or DNA, and mostly would be DNA immobilized on the surface of the biosensor. And this DNA strands will bind to complementary sequence, and binding of the complementary sequence or the target complementary sequence will result in a signal that will be detected by the transducer. Also, we can use an enzyme immobilized on the surface of the sensor, like in case of glucose biosensor, which will be able to recognize a specific substrate in the biological specimen. Again, binding of the substrate to the enzyme will result in a signal that is sensed by the transducer. And another example for the biological or the biosensing element will be an antibody immobilized also on the surface of the biosensor, and this antibody will detect a specific analyte in the biological specimen. Binding of the analyte to the antibody will result in a signal that is sensed by the transducer. طبعا يعني احنا بنقول بسهوله كده ان we can use an antibody immobilized on the surface of the biosensor, and this antibody will be specific to a specific analyte in the blood of the patient, مثلا, for example. But طبعا there are many strategies for the use of antibody for detection of a specific analyte. So in different electrochemical immunosensor, we can use a capture antibody that will detect or bind to an analyte in the blood of the patient, and then you can use a detector antibody with a label to bind to the antigen, and this will result in a signal that will be detected by the transducer. Or we can use a capture antibody to bind the target analyte or the target antigen in the blood of the patient, but also we can add a labeled antigen which will compete with the endogenous antigen for the capture antibodies, and this will result also in a signal that is sensed by the transducer. On the other hand, a third strategy will be to immobilize the antigen itself on the surface of the biosensor, and then we use antibodies labeled with a label in order to detect the presence of the antigen, and also this will result in a signal that will be sensed by the transducer. Again, you can see here, this is my capture antibody, this is my analyte, this is my detector antibody labeled with enzyme, and this enzyme can convert a colorless substrate, for example, into a colored substrate or electroactive product, and these electrons will be sensed by the transducer, converted into a signal, and the signal finally will appear on the screen on the biosensor. So even if you select an antibody to detect the antigen of interest or the analyte of interest in the blood, there are many strategies. By the way, this is only a limited number of the possible strategy that can be applied or implemented when using the antibody for detection of analyte of interest in the blood. The same thing, instead of using an antibody for detection of the analyte, you can use an aptamer. And as we know, aptamers are DNA sequences that will take a 3D structure, and this 3D structure will be able to detect a specific analyte in the blood or any other biological specimen by what we call molecular recognition or biomolecular recognition. Again, there are many strategies. So even if you decide to use an aptamer as your sensing biological moiety in the biosensor, you can use many strategies. And here also we only list 8 different strategies for the utilization of aptamer for detection of a specific analyte in the blood. For example, here you can use a molecular beacon with a fluorescence and quenching molecule next to each other, but then the presence of the target analyte will result in the opening of this molecular beacon, and then the fluorescence molecule will be kept away from the quencher, and a fluorescence signal will be detected. Here we have an aptamer also with a fluorescence molecule, but the fluorescence here is quenched by the carbon nanotubes in this particular strategy, and presence of analyte will lead to the binding of the aptamer to the analyte, and this will keep the fluorescence molecule away from the carbon nanotubes, and this will result in the detection of a fluorescent signal. In this case here, we have the aptamer on the surface of gold nanoparticles, which are red in color. Presence of the analyte or the target molecule will lead to binding of the aptamer to the target molecule, and they will depart from the gold nanoparticles. In this case, the color of the solution will change from red to blue. And again, we have many other strategies that can implement the aptamer as the biological sensing moiety for detection of the analyte of interest. We have 4 different types of transducer. So we can have an electrochemical biosensor. In this case, we have electrical signal that will be detected either in conductometric or potentiometric or amperometric manner. We have also mass-based biosensor, in this case, like piezoelectric and acoustic wave. We also have the calorimetric biosensor, which detect heat. And we have the optical biosensor, and the signal here will be colorimetric, interferometric, fluorescence, or luminescence. So in a way, for example, in optical biosensor, binding of the biological moiety in the biosensor to the target analyte, in case of colorimetric biosensor, will result in a change of color. And this color will be detected, and this will indicate the presence of the target analyte. In case of the electrochemical biosensor, in this case, binding of the biological moiety in the biosensor to the target analyte will result in an electrical signal which can be detected and displayed on the screen of the biosensor. What are the criteria of a successful biosensor? The biosensing element or the biological entity used for recognition of the analyte must be highly specific, must be stable under normal storage condition, and must provide reproducible data, which is important for any diagnostic test. Recognition of the analyte by the biosensing moiety should be independent of all other physical parameters like pH, temperature, and steering, and so on. And the response of the biosensor should be accurate. The biosensor must generate results rapidly since it is required for real-time analysis, and the biosensor itself should be small in size in order to be portable, should be inexpensive, because being inexpensive will allow a larger portion of the patient population of accessing and acquiring this biosensor. And then finally, it should be easy to use and can be used by non-skilled operators, very similar to the glucose biosensor, which can be used by elderly people because it's very simple and very straightforward to use. I will give you now examples of different biosensors. The most famous biosensor are the amperometric glucose sensor. Actually, it represents over 80% of the biosensor market. زي ما احنا عارفين, and the biosensor market now is estimated at almost $30 billion industry, 80% of which is for glucose sensors. طبعا الجلوكوز سنسور الفكره بتاعته the biological sensing layer or moiety in the biosensor is an enzyme which is glucose oxidase. So glucose oxidase in the biosensor will convert glucose into gluconolactone in the presence of cofactor FAD, and this will result in generation of electrons which can be received by oxygen, converting it to hydrogen peroxide, and then this will result in and this will be sensed by an electrode. In the second generation glucose sensors, a mediator was used instead of the oxygen in the blood to receive the electrons from the oxidation reduction reaction of the glucose oxidase. And these mediators may include ferrocyanide, ferrocene, or methylene blue. And this will deliver the electrons from the redox reaction to the electrode. Another generation was 2.5, the generation of the sensor. They immobilize the mediators on the surface of the electrode. And then finally, in the most recent and the most advanced third generation glucose sensors, the redox reaction of glucose oxidase to convert to glucose to gluconolactone will result in electrons which will be directly transferred to the electrode and will be displayed on the screen of the biosensor. Most recently, researchers have reported the development of NI glucose biosensor. In this case, the biosensor will be inserted in the lower lid of the eye, and it will measure glucose in the tears of the patient. And there have been several reports showing very good correlation between levels of glucose in the tear and also in the blood. Again, this sensor is based on the use of the enzyme glucose oxidase, which will convert glucose into gluconolactone in presence of fat, and this will result in release of electrons, which will be sensed by an amperometric electrode. Also, lactate can be sensed using lactate sense biosensors, and this is actually can be applied as a tattoo on the arm of the athlete who is exercising. And these biosensors essentially contains the enzyme lactate dehydrogenase, which can convert lactate into pyruvate, and also this will result in presence of NAD, and this will result in generation of electrons, which will be sensed by the carbon electrode. Again, these biosensors will have biofuel cells and should be self-powered biosensors. Again, they will measure the level of lactate for this particular person who is exercising, and then will alert him by sending a signal to his mobile phone that levels of lactate are going up, and in this case, he will need to stop before he starts suffering from muscle cramps and so on. The biosensor have been integrated with microfluidic devices and also smartphones for point-of-care testing of different disease and physiological biomarkers. For example, in this case, we can have a microchip that can detect different analytes, maybe can detect multiple analytes in the blood of a patient or saliva or urine sample, any kind of biological specimen. And this microchip will be inserted in a mobile phone for data to be analyzed and be displayed, as well as being sent to the medical the doctors who are following this particular individual. Also, the mobile phone may have an optical sensing attachment, by the way, which can be used as a sensing device in order to detect the signal in the microchip. And in this case, the mobile phone will act as a detector, and also it will perform data analysis and display of data, and also sending the data to the indicated facilities. This is an example of a coronavirus smartphone detection device. So in this case, we have a sensor that will be plugged into the smartphone, in the charging port of the smartphone. And then the person in this, of course, an application will be launched. And then the person using this particular phone will either cough or sneeze or blow on the sensor. And then if there are coronavirus in this particular individual, a signal will be generated, either positive or negative, to indicate whether or not this person has a coronavirus. And at the same time, the application will update the coronavirus map. So health authorities will understand whether or not a particular area in the city have multiple persons with coronavirus, because this could be a focus of infection, in order for health authorities to intervene with this particular area. طبعا كلنا سمعنا عن ان Nobel Prize in Chemistry this year has been awarded to Emanuela Charpentier, as well as from Max Planck Institute in Germany, as well as Jennifer Dodana at University of California, Berkeley in the US, for their work on the CRISPR technology, or the gene editing technology. As we know, CRISPR is essentially, we have the Cas13A enzyme, and this enzyme has two RNase activities: RNA recognition and cleavage, as well as we have a guide RNA of CRISPR RNA with a particular sequence that is complementary to the target sequence, for example, for the RNA of the coronavirus. In the presence of the RNA of the coronavirus, the CRISPR RNA will bind to it, and this will activate the Cas13a to do what we call cis cleavage of this particular sequence. But then the Cas13a enzyme will perform what we call transcleavage of multiple RNA sequences. In this case, we have reporter molecules, which are other RNA sequences with quencher and fluorescence, and their cleavage will result in the release of a signal that will indicate the presence of the target. So in this case, the CRISPR system will be extremely sensitive, because the presence of a single copy of the virus RNA will activate Cas13 enzyme in order to do cis cleavage of this particular copy. But then the Cas13 will also carry out what we call transcleavage of many copies of the reporter RNA molecules, which will result in the generation of a large fluorescent signal, which indicate the presence of the virus. So again here, the Cas12a enzyme will once the target sequence is present, it will bind to it and then will do cis cleavage of the target, but also it will perform a transcleavage of non-target sequences, which are the reporter molecules. In this case, the reporter molecules contain methylene blue, and this will result in a signal which will be detected, indicating the presence of the target. As I mentioned, in this case, a single copy of the target is enough to generate a strong signal indicating the presence of the target, the RNA molecule. This is my last slide on mouse biosensor. This is a very interesting biosensor developed for detection of COVID-19 or SARS-CoV-2. It's called the RapidPlex. Again, it's a very straightforward. This is a screen printed electrode. So a screen printed electrode has 4 different dots. In the first dot, we have an antibody which will detect certain proteins on SARS-CoV-2 or the COVID-19, and the presence of this antigen will indicate that this patient actually has COVID-19 virus or SARS-CoV-2. So this indicates, so a positive signal in this dot will indicate infection with COVID-19. In the dot number 2 and dot number 3, we have here antigens which can detect the presence of antibodies made by the body against COVID-19. But the first dot here will indicate the presence of IgM or immunoglobulin M molecule, and the third one indicate the presence of IgG or immunoglobulin G molecules. So this dot number 2 and dot number 3 indicate immunity of the patient against COVID-19 and the presence of IgM and IgG. The final spot has antibodies which can detect the C-reactive protein, which is a marker of inflammation. In this case, this spot really indicate or assess the severity of the disease of COVID-19 because it's a marker of inflammation. So using a single biosensor, we can detect the 4 different analytes. In this case, the antigen of the virus itself, which indicate infection. In spot number 2 and 3, we can detect antibodies, IgG and IgM, which indicate immunity of the person against the virus. And then we can also assess severity of immunity by measurement of the C-reactive protein, which is a marker of inflammation. This is my last objective, or my last objective for this lecture, which is giving you a roadmap if you were to start a biosensor research, or if you want to start your biosensor research and you want to develop your own biosensor. Number 1, you have to select the disease biomarker. يعني what are your biomarker of interest? example, مثلا, do you want to develop a biosensor against COVID-19? So in this case, you need to decide on which analyte. Are you going to detect a protein from the virus or the virus RNA itself, or an antibody made by the body against the virus? So in this case, you have to decide on the disease biomarker. This will be step number 1. Number 2, what kind of specimen, what kind of biological specimen do you want to detect this analyte in blood, or in saliva, or in urine, or any other kind of biological specimen? And in this case, you have to decide whether or not you need to do a sample pre-treatment or clean up the sample before you apply it to your biosensor. then you need to decide whether or not you need what we call a qualitative biosensor or a quantitative biosensor. يعني is مثلا in case of COVID, it is really not important to decide to determine the number of viral particles of COVID-19 in the blood in the patient. What's important is to know whether or not the patient has COVID-19. So in this case, we are interested in a 0-1 answer or a binary answer. Yes, the COVID-19 is present, or no, it is not present. Or do you want a biosensor that can generate a quantitative answer? In this case, like glucose biosensor. يعني طبعا كلنا عندنا glucose, بس احنا we need to know is glucose level is too high for 500 مثلا milligram per deciliter whatever, and this will go on hyperglycemia, or is it too low? Because in this case, so in this case we need a quantitative answer. Of course, we need a calibration curve that will be stored in the biosensor, and in order to calculate the level of the biomarker in the blood. Also, we need to select the biological recognition moiety. Are you going to use an antibody, or going to use an aptamer, or what you're going to use to detect your analyte of interest? You need to select the transducer. What kind of signal will be generated when your biological recognition moiety binds to the analyte? Is it going to be a colorimetric signal or electrical signal, and so on and so forth? In this case, you also need to worry about the data display. Will be displayed on the biosensor itself, or the data will be displayed on a mobile phone or on a computer, or will be just simply be sent directly to the hospital or the medical or the healthcare facility. Finally, the analytical performance of the biosensor should be assessed with regard to its specificity, sensitivity, negative predictive value, positive predictive value, and accuracy. And then the reproducibility of your biosensor. Finally, you need to worry about data liberation by using Internet of Things. It is of no use to keep the data in the biosensor. I'm interested for this data to reach the medical facilities in order for a medical decision to be taken and intervention with the patient or the person if necessary. I thank you very much for your kind attention, and I will be happy to answer questions. I will call the Dr. Tariq Abul Fadl by phone after my lecture in order to receive any questions, and inshallah I'll be able to answer them. And thank you very much for your attention.
احنا بنشكر الاخوه المحاضرين على المحاضرات القيمه والتي استفدنا منها كثيرا واستفاد منها المستمعين. احنا يعني بس الدكتور محمد عباس لنا مجموعه تساؤلات. الدكتور محمد عباس سمعني؟
سامع حضرتك يا دكتور، يا مرحبا.
طيب يعني سلامنا للاخ العزيز ابو مالك الدكتور فاشل عنب، بلغوا سلامنا. الدكتور عبد الظاهر معاك.
الله يسلمك.
بس في شويه سؤال يا دكتور محمد، يعني ايه البيزس اللي انت اخترت عليها البوليمرز اللي بتستخدمها في الكومبريشن اوف نانو بارت؟
طبعا يا دكتور مش كل البوليمرز سيوتابل انها تكون ستابلايزر للنانو بارتكلز.
ايه البيزس بقى؟ ايه البيزس بتاعت البوليمرز ده؟
طبعا هم يعني الانفستيجيتورز عملوا سكريننج لمجموعه من البوليمرز، ولقوا ان البوليمرز اللي ليها الابيليتي تو اتاتش تو ذا سيرفيس اوف نانو بارتكلز تو ميك از بريدجنج. في نوع من الستابلايزرز، في ستابلايزرز are acting by bridging، يعني attached to the surface of each nanoparticle and making bridging. ودي مش كل البوليمرز، يعني مثلا البوليثيلين جلايكول ملوش القدره دي الكبيره انه يعمل كده، لكن البوليفينال بيرودون، البولورونيك، التوين فور اكزامبل ليهم. في انواع ثانيه من الستابلايزرز يا دكتور يعني based on the formation of electrical charge around the formed nanoparticles، وبالتالي بتزود الريباركشن بين النانو بارتكلز، وبالتالي تمنع النانو بارتكلز to be close to each other. يعني انا مش سامع دكتور الصوت كويس.
كريستالين فورم. طبعا انت عارف ان لما تحول الدراج من امورفس كريستالين فورم ده ويل افكت البايوافيلابيلتي، هينقص البايوافيلابيلتي. يعني ايه تفسيرك الحكايه دي؟ يعني المفروض ان النانو بارتكلز يعني ويل انكريس البايوافيلابيلتي ما بتوصلش دراجز، فانت دلوقتي بتقول ان may some preparations will convert the amorphous state of the drug to crystalline form.
نعم.
How can you counteract this يعني؟
لا حضرتك انا ذكرت ان one of the issues of nanoparticle stability is the crystal growth. يعني النانو بارتكل حضرتك دي عباره عن very minute particles has or have high surface free energy, so they tend to be close to each other to minimize this surface energy، وبالتالي بيحصل aggregation وبيحصل crystal growth. دي من ضمن العيوب بتاعه النانو بارتيكولت سيستم يا دكتور. وبالتالي
how can you counteract this conversion؟ يعني انت دلوقتي بتقول انه في البريبريشن بتحول الكريستالين فورم.
نعم.
معمول الكريستالين فورم البيبرابيلتي بتاعته اقل من الامورفس.
لا حضرتك انا قلت ان هو during storage one of the nanoparticle problem او instability problem is the formation of or increasing of the particle size and increasing of crystal growth. بيسموها الاوستفالد رايبنينج او اوستفالد جروث نتيجه الاجريجيشن اوف nanoparticles to lose their surface free energy. علشان كده دي عيوب، دي احد عيوب النانو بارتكل دراج ديليفري سيستم.
طيب الاستفسار الثالث انت بتقول انه الستيرك هندرانس او الستيرك ستابلايزيشن بتاع النانو بارتكلز والريديوس تشارج such بارتكل. الكلام ده طبعا هيؤدي الى ان الزيتا بوتنشال اوف ذيس بارتكل ويل ديكريز، وانت عارف ان القيمه بتاعت الزيتا بوتنشال لازم تبقى ماينس 32 عشان يبقى ستيبل. فانت دلوقتي نقصت التشارج موجود على البارتكل، ومن ثم هذا يؤدي الى انستابلايزيشن.
As I mentioned before يا دكتور ان we have two types of stabilizers. One of which is acting by bridging between nanoparticles، وبالتالي دي مجموعه البوليمرز البولورونيك والبي في بي والاتش بي ام سي والمجموعه دي، وفعلا في هذه المجموعه بيحدث ريديوسنج لزيتا بوتنشال نتيجه البريدجنج اللي بيحصل بالنانو بارتكلز، لكن ما بنوصلش ان نعمل يعني very high reduction of the zeta potential.
هو بيحصل ما دام هو بيحصل ريدكشن للتشارج، الزيتا بوتنشال بتعتمد على الريبالشن اوف ذا ستارش اوف ذا موليكيول او صح؟ فكده يعني لو نقصت ازيد من اللازم بهذا الخصوص يعني ده بيؤدي الى decrease في الاستابيليتي اوف ذا سيستم.
ده صحيح وعلى ذلك حضرتك احنا بندرس الايفكت اوف concentration of the polymeric stabilizers on the zeta potential to يعني to attain suitable zeta potential زي ما حضرتك بتقول، يعني not exceeding minus 32 او not less than around minus 13 or minus 12 ميجا ملي فولت.
طيب السؤال الاخير، الابحاث قيمه الحقيقه وشغل ممتاز يعني، بس هل في بريبريشن من نانو بارتكلز دي بريزنتد في الماركت ولا كله؟
لا حضرتك يا دكتور انا عرضت اكثر من 8 formulations. One of them is based on the hydrophilic nanoparticles اللي هو based on the albumin اللي هو الابريكسين اللي فيه الباكليتاكسل ده ماركتد برودكت اوريدي. في ماركتد برودكت ثانيه based on the لايبوزوم يعتبر نوع من النانو بارتكل اللي هو الدوكسوروباسين كانتي كانسر دراج. في مجموعه من الفينوفايبريت، بعض الشركات انتجت فينوفايبريت based on the nanoparticle drug delivery system، ودي بتتحضر بالتوب داون ميثود. في بعض انواع ثانيه بتتحضر بالبوتوم اب ميثود، ودي موجوده اوريدي في الماركت يا دكتور، حوالي 10 ماركت برودكتس.
الدكتور اكرام عايز يسالك.
اوكي انا شكرا دكتور محمد. انا عندي سؤال معلش هنتقل للبريزنتر تاني عشان نغطي الاسئله كلها. سؤالي للدكتور محمد حسن. Question about first, thank you very much for your interesting presentation about MI31 and its role in gynecological cancers. And my question is about what if this MIR is epigenetically controlled?
Well, thank you very much for your nice question, Dr. Akram. In fact, as you could see in my presentation, we just introduced a series of evidence that MIR31 is a key player in the chemo response. So we have now evidence that if this expression had been restored, of course, the chemo resistant, especially to Taxol, will be disturbed. That's why we thought about the optimal mechanism. لكن في الحقيقة, even if this one had been downregulated due to epigenetic reason, of course, that we'll switch our gears to think about the epigenetic inhibitors, which already established. So we can combine the mere overexpression together with the epigenetic modulator like HDAC inhibitor, for example, or even DNA methyltransferase inhibitor, which already in clinical application. Thank you very much.
Thank you very much, Dr. محمد.
طب انا ليا سؤال للدكتور محمد برضه.
اتفضل.
حضرتك المايكرو ار ان اي 31 is concerned with gynecological cancer. Why not to other cancer؟ يعني انت حضرتك حددته للجينيكولوجي.
هو حضرتك اكزاكتلي انا كنت متعمد لان احنا يعني most interesting ان هو it shows different totally opposite function in these two types of gynecological cancer. However, from other groups, of course, we could see that it has some role. Of course, not all of them have been studying different cancers, especially in chemo-resistant. لكن بالفعل, for example, in some types of cancer, like as far as I remember, osteosarcoma, for example, it had been truncated with some from some chromosomes.
يعني ده ممكن يعني.
بالفعل different function. هو بصراحة ده المسج يعني تيكو مسج أنا عايز أقولها إنه الفانكشن is different according to the tissue. That's it.
طب السؤال الثاني انت حضرتك طبعا يعني عملت induction للجينيكولوجيكال كانسر to the experimental animal مش كده؟
لا الحقيقة ده كان transplantation مش induction. It's a kind of tough يعني job إن إحنا نعمله. However طبعا هو ممكن لكن
experimental animals.
في experimental animal what I did is transplantation. I mean xenograft model. نود مايس.
يعني انت دلوقتي حضرتك مش اختبرت هذا الكلام على كانسر في الجينيكولوجيكال سيستم في الانيمالز؟
مش بالظبط يا فندم. هو انا حضرت السيلز بتاعتي وعملت لها ترانسبلانتيشن في نوع من المايس اللي هو النود مايس اللي هو بيتحمل الهيومن كانسر سيلز. وبعدين لما الايفيدنس هاد بين كونفيرمد سويتشت بقى لكلينيكال سامبل من المستشفى هي هيومن اوفرين كانسر ودي اللي اكدت فيها الفينومينا.
طب هل البريبريشنز دي يعني اي منها يعني ترانسميتد تو الفارماسيوتيكال ماركت ولا جاست انفستيجيشن؟
اعتقد يا فندم دلوقتي هو يعني ستيل اندر بري كلينيكال ستادي اللي هو الكايميريك ابتمر اللي انا قدمت ليه في الاخر اللي احنا حاولنا نوصل له كابلكيشن. كايميريك ابتمر ده اوريدي في البري كلينيكال ترايل حاليا في الانيمال مودلز.
شكرا يا دكتور محمد على المحاضره القيمه.
Dr. Muhammad, I have another question. I'm very sorry, but I'm talking about if this MIR31 is administered systemically, what do you think about the stability and how it can reach to the tumor tissue?
Well, I can understand the point you came from. I think you are thinking about the delivery and the mechanistic, right? However, what I can say from my today's talk, it's almost difficult to be administered systematically because as you could see even if some patient with ovarian cancer and I could deliver, of course, will cause damage or maybe cancer in case of endometrium. That's why it's totally difficult to say yes. However, of course, specific delivery like the aptamer I showed could be the best solution, I guess.
I think I can help you to target it systemically. So let's collaborate about it.
It's my pleasure. It's my pleasure.
I have a question for. Thank you, Dr. Muhammad. I have a question for Professor Azazi. You talked about the detection of COVID-19 by using the biosensor, depending on CRISPR. Do you think this is cost-effective compared to the available PCR detection system? And how about, I think the accuracy is high, but how about the cost?
Thank you very much for the question. I think the issue with the biosensor, it will give you an almost immediate answer. So the time here will be very important. Like right now, even if you are doing a nasal or nasopharyngeal swab and then do PCR, you need at least 24 hours if you have an expedited surface, or maybe up to 48 hours to generate the results. Sometimes in airports and other places, you need an immediate answer. So something like this, a biosensor, even if it's a little bit more costly, which I doubt it will be more costly, because once we produce the enzyme Cas13a and the guide RNA for this should not be very, at a larger scale, of course, this will not be very costly. Okay. But the importance here will be the sensitivity of the assay, as well as, because here we're talking about a single copy of RNA of the virus, which can be cleaved. Exactly.
I really wanted to hear this because this is very promising to be applied.
That's right. This is really promising. Yes, thank you.
دكتور حسن، حضرتك قلت الميجر الجلوكوز باي اي سنسور هل دي براكتيكال ميثود انك انت تقيس الجلوكوز in the eye by a sensor؟
This is a trend actually, and actually Google has developed its own Google glasses. So these are contact lens that can be placed in the eye and also measure glucose and other biomarkers in the tears. As I mentioned in my presentation, there has been several studies. Right, it is complicated. It is complicated because the design of the contact lens will have to take into consideration several parameters. The curvature of the eye, it should not interfere with the vision itself. It should have some sort of its own battery to generate the data, because it cannot, and also it has to have some sort of an antenna to transmit the result from the glasses to the system that receive the signal from this sensor. So it's technologically challenging. I doubt very much in Egypt we can do something like this. It's very advanced. It's funded by Google. It's called a Google X company. But I think for us here in Egypt, and this is why the purpose of my presentation is to provide some sort of a roadmap or some sort of a flow chart for anyone or any research group in Egypt who want to develop a biosensor. It's a very exciting area of research. There are many possibilities. It can be developed. In my research group here at AUC, we develop several biosensors. We participate annually in the International Biosensor Competition in Eindhoven. So I think this is a very important and very promising area of research that I want to see really more research groups in Egypt and national universities engage in this kind of research.
Thank you. طيب آخر سؤال، الكريسبر تيست اللي حضرتك ذكرته على الكورونا فيروس، هل حضرتك عملت له ابلكيشن في
No, we just submitted a grant on this technology in order to develop a CRISPR-based test, but we have not really initiated this. We have several other biosensors that we have developed in my group, and also we have patents actually in some of these. We have patents on chemosensors, not biosensor, also in my group. But we have not yet initiated the work on CRISPR. But it's again very promising.
Okay. دكتور محمود الصباحي معنا؟ مش موجود؟ معنا ولا مش معنا؟
لا مش معنا.
طيب احنا بنشكر الاخوه المحاضرين على المحاضرات القيمه، وسعدنا بكم كثيرا، وان شاء الله نلتقي في المؤتمرات القريبه باذن الله. السلام عليكم ورحمه الله وبركاته.
وعليكم السلام ورحمه الله. شكرا جزيلا.